The reigning scientific consensus on human consciousness relies on the premise of strong emergence. Philosophers like Daniel Dennett and the majority of modern neuroscientists operate on the belief that subjective experience is generated entirely by the sheer complexity of biological neural networks. We are examining a strictly bounded metaphysical architecture that challenges this premise. This is a logically coherent framework for reality, presented not as a proven law, but as a formal alternative to the materialist generator model. This split screen illustrates the core thesis. On the left, the standard model. The brain constructs consciousness from the inside out. On the right, the substrate brain framework. An ambient frequency field passes through a 3D brain, which acts as a prism. In the first model, the biological hardware acts as a generator. In the second, it functions as a translator. This frequency field originates within a four-dimensional, inert substrate, a passive ground state underlying all localized space-time geometry. This architecture refines the transmission theory famously articulated by William James. He argued the physical brain restricts and channels a pre-existing field of awareness, rather than creating it from neural activity. Treating the brain as an antenna relocates the hard problem of consciousness. It moves the brute fact of experience away from biological hardware and into the structure of the cosmological substrate itself. If consciousness is an ambient feature of the substrate, we must explain the combination problem, why individual human experiences remain distinct rather than blending into a single overlapping field. The framework identifies DNA as the source of a bespoke biological tuner that prevents this overlap. This mechanism draws on the orchestrated objective reduction theory proposed by Sir Roger Penrose, which posits that quantum computations occur within the structural proteins of neurons. This tuning happens far below the level of neural networks, within the cytoskeletal microtubule network of individual cells. DNA dictates the assembly of these microtubules, arranging the tubulin proteins into a unique geometric lattice. This physical geometry determines a highly specific resonant frequency, the exact and only frequency that a particular brain is capable of receiving. This structure protects a quantum coherent state from the warm, wet environment of the brain, allowing it to maintain a stable connection to the substrate. The lattice vibrates at a mathematically precise frequency, creating a unique signature that defines a distinct individual conscious identity. This resonance is highly exclusionary. Because the substrate signal is non-local, only a biological receiver vibrating at the exact mathematical reciprocal can pull that specific pattern down into three-dimensional space. Any variation in the tubulin geometry results in a frequency mismatch. This mathematical exclusion ensures that one consciousness pattern remains bound to one specific body, solving the combination problem through resonant specificity. With the individual Qualton lock established, the macroscopic brain begins its primary task, the translation of the substrate signal into localized experience. This model shows an incoming signal from the substrate interacting with the distinct functional networks of the cortex. These specialized hubs convert the raw signal into the specific perceptions and behaviors we recognize in three-dimensional spacetime. While the incoming substrate signal is completely continuous and unified, the biological networks translating it are highly modular and compartmentalized. In this framework, neural activity does not determine the presence of awareness. It dictates the specific flavor, limits, and sensory content of that awareness. Our perception of the world is not the raw signal itself, but the mediated result of that signal being translated by biological hardware. We can test this translator model against clinical lesion data. Historical cases like Broca's and Wernicke's aphasias clearly show that focal brain damage reliably alters specific cognitive and perceptual functions. The central question is why localized damage can radically warp the content of a patient's reality while their core sense of awareness persists. The substrate brain framework treats these symptoms as localized mistranslations. The physical damage disrupts the hardware's ability to correctly process the incoming signal, but it does not extinguish the signal itself. Consider the Capgras delusion. This syndrome relies on two distinct processing nodes, one for visual recognition and another for emotional tagging. In a healthy brain, the signal passes through both nodes, producing a harmonious output wave experienced as a sense of familiarity when looking at a loved one. In Capgras syndrome, the connection to the emotional tagging node is severed. The signal passes through the intact visual node, the face is recognized, but without the emotional tag, the final translated output becomes distorted. The patient experiences a cognitive glitch. The person looks like their spouse, but feels entirely wrong. They conclude that the person must be an exact imposter to explain the mistranslation. Materialist models rely on auxiliary hypotheses to explain how brain damage can generate such highly specific, coherent delusions from missing data. Under this architecture, focal damage acts like a flaw in a prism. It distorts the resulting colors of experience while leaving the fundamental light source unaffected. This framework provides a specific interpretation of the mechanics of death, the moment when the biological tuning hardware fails completely. This transition is visible in the empirical data recorded from brains at the moment of clinical death. This chart shows a flatlining heart rate overlaid with a sudden, massive surge in high-frequency gamma oscillations, a phenomenon documented in multiple human and animal studies. The mainstream materialist view interprets this surge as a final, last-gasp network dynamic as oxygen is depleted. The substrate brain framework identifies this gamma spike as the rapid, chaotic loosening of the DNA-governed quantum tuning lock. The surge in electrical activity marks the final failure of the biological antenna. It is the moment the hardware loses its grip on the frequency, allowing it to release back into the substrate. The framework posits that the post-mortem trajectory depends on wave turbulence, the degree of coherence the conscious pattern maintains at the moment of separation. A perfectly coherent, stable waveform achieves a tuned merge, integrating quietly back into the ground state of the substrate. High turbulence, resulting from trauma or mental instability, leads to untuned drift or stuck loops, where the pattern remains fragmentary and unable to integrate. We must acknowledge the Popperian limits of this model. Because the four-dimensional substrate cannot be measured with three-dimensional instruments, these terminal outcomes remain scientifically unfalsifiable. While not a proven empirical law, the substrate brain framework offers a mathematically compatible architectural schematic. It provides a unified explanation for anomalies that the materialistic generator model cannot yet account for.